morning, for today, actually. And I think we have, Nina needs to be promoted. And it's about fluorescence and fluorophores. So let's see, what is fluorescence? So fluorescence is a cycle of photon absorption and emission. So it's a cycle. The fluorophores are going back and forth between being excited and being not excited. Only some molecules can fluoresce. So they're called fluorophores or fluorochromes. Sometimes fluorochrome is just the part of the molecule, it's called just a part of the molecule that can fluoresce, but you can call it fluorochrome, fluorophores, it's a little bit indistinguishable. An excited fluorophore acts like a light source in the sample. So we have lots of little light sources in our sample. So this is what it can be represented as. And we have, this is the molecule. We have electrons around the nucleus of the molecule, and a photon comes in from the excitation light. And you can see in here that we have this photon is in the UV, so it's sort of color-coded in purple. And the electrons get excited and they go into a different state and they change their behavior. But there is like a lot of vibration going on in the molecule. And so the molecule loses a lot of heat. And despite this, it doesn't lose all its energy, but there's a loss of heat in vibrations, and the leftover, the energy, makes the molecule anyway unstable. And so the molecule is going to spit out the extra energy in forms of light. So it gets rid of the energy in forms of a photon. And because we have these vibrations between the excitation and the emission, then the emitted light has a lower energy than the excitation light most of the time. So this is the way we can represent this. Here we have the light that is absorbed by the fluorophore, and then in here we have the emitted light, and you can see that there is a shift between the peaks here, and it's called the Stokes shift. Maybe some of you have heard about this. And what is it? It is basically the difference in wavelengths between the peak of the excitation light and the peak of the emitted light. So there is a loss of energy, and therefore the light that is emitted is more towards the red than the light that is used to excite the sample. This is the case most of the time, not always. So another way to represent these excitation cycles is like this. This is called the Jablonski diagram, and it's presented very often like this. So we have the different states of energy at 3 different levels. In here, this is called the ground state, and you can see that we have anyway some variations. So in this state, the molecules can be relaxed, so they are not excited, and they are here in this ground state. And then what happens is that when we shine some light on it, the fluorophore will become excited, and it goes up into this singlet state. And in the singlet state, the molecule is unstable, and therefore they're going to vibrate and lose some energy and go into yet another state. Okay, so from that intermediary state, several things can happen. One thing that can happen is clearly the fluorophore can turn off without emitting light. That can happen if it vibrates enough. That's one possibility. Another thing that can happen is that it will emit some light, and there it will go back to the ground state after emitting some light. Something else that can happen is that it goes instead into the dark state, which is also called the triplet state. And when this happens, the molecule is not fluorescent. So it is excited, it has acquired, let's say, the energy from the excitation light. So it's excited, it is stable, but it does not fluoresce. So this is why it's called the dark state, okay? So it's different from this place where the molecule is excited, it's unstable, and then it may fluoresce. So what happens here in the dark state is that the molecule can stay there for a little while, and then after a while, the same thing happens as the other one. Maybe it will vibrate enough that it will go back to the ground state, but maybe it will actually emit fluorescence, okay? So we have these different states of energy in a fluorophore. Now, there are also some things that are sometimes confusing, and it's all these things about luminescence versus phosphorescence and fluorescence. So luminescence is all phenomena where light is emitted, and that includes fluorescence. Chemiluminescence is the same thing, so light is emitted, but the trigger is not light excitation. The trigger of light being emitted is a chemical reaction. And so bioluminescence is all type of luminescence that is found in nature. So for example, fireflies and corals. So all these terms really refer to the same thing, but depending on where it is, and also if it's chemical or it's triggered by light, then we have slightly different names. And then we have phosphorescence and fluorescence. And in that case, the luminescence is triggered by light. And the difference between phosphorescence and fluorescence is that in phosphorescence, the molecules first go into this dark state, and then they stay there for a while before they emit some light. And therefore, there is some sort of a delay between the excitation and the emission. So this is phosphorescent. Basically, it takes much longer for the molecule to start fluorescing because it gets stuck in the dark state. The transition here from the excited state to the dark state is a statistical process. And so that means that whenever we image some fluorophores, some of them will go into this dark state, whatever we do. But we have some tricks that we can use to make it go faster into the dark state if we want to. I will explain why. And we have some tricks also to prevent it from going to the dark state. But there will always be some molecules that will go to the dark state. When we shine a very strong light for a long time on fluorophores, because the transition to the dark state is a statistical process, there's a higher risk, let's say, or there's a higher proportion of the molecules that will get trapped into the dark state. So we can, by shining strong light for a long time on fluorophores, we increase the risk for the fluorophores to go into the dark state and get stuck there. And what happens is then that they will not fluoresce. So it will look like our sample is less bright. So when we image on a microscope, when we image with a high illumination power, and this is especially the case for single point confocals that most of you use, when we image with a high illumination power, it is important to try and image for a short time as much as possible. So it's better to image often and for a short time than image once very, very slowly. So, when you set your microscope, you know that you can change when you set your point confocal, you know that you can change the speed of the scanning. You want to avoid using a long speed, like a slow speed and a long pixel dwell time, it's called, because then you have a higher risk to send your fluorophores to the dark state, and then it looks like your sample is bleaching, even though it isn't. So on single point confocals, try to use a frame averaging instead of a slope speed. Or we will talk about all these different settings later. But this is the consequence of us having this thing that is called the dark state. What happens next is that the fluorophore, when it's excited, it can also photobleach. And it can create photo damage. So once the fluorophores are excited and they go from the ground state to the excited singlet states, they are very unstable there. When they are in this state, they meet an oxygen molecule. Then there is an irreversible destruction of the fluorophore, and this is bleaching. So this is irreversible, the fluorophore is gone, it will not come back, okay? But there is something else that happens. At the same time, we have the production of reactive oxygen species. And these oxygen species are going to diffuse everywhere very quickly in the sample. And that leads to heavy photo damage for the sample. So we want to try and avoid photo bleaching because we also create photo damage. And that is the destruction of or the modification of macromolecules. For example, DNA can be modified with photo damage. So we want to be very careful with not having too much strong light on our sample. And now, how can we prevent this reaction with oxygen in the medium? We can use some chemicals, so oxygen scavengers, also called anti-fade agents. So this is the type of things that we have in our mounting media. the oxygen scavengers will prevent the oxygen from destroying the fluorophore and creating reactive oxygen species. We can also physically prevent oxygen from coming into the microscope by adding a cover slip on top of the sample and sealing it, and that prevents oxygen from reaching the sample. That also works very well into preventing photobleaching and photo damage. This is for samples that are in medium, like live samples, for example. And you can see here that the bleaching is not linear. So bleaching goes much faster at the very beginning and then it slows down. So it is really important do not look at fluorescence in the eyepiece if you can avoid. In this course, we will teach you how to find your sample and focus on your sample without looking at fluorescence in the eyepiece. Okay, so this is a technique that is very simple. It works for all microscopes. And once you get used to it, you will not ever need to look at fluorescence in the eyepiece. Either you focus on your sample with transmitted light or you image it, but you don't waste it by looking at fluorescence in the eyepiece because then you bleach it. Okay, but so now in this the second part of this tiny lecture, we have two types of fluorophores. So the fluorophores that are produced by chemistry and the fluorophores that are produced by cells. So by chemistry, they are chemical fluorophores. They are called dyes, and but otherwise, those that are produced by cells are proteins. And we also have autofluorescence in the samples. They're also produced by cells, and these can be proteins, but they can be also macromolecules, metabolites, pigments, all sorts of things that can fluoresce in our samples. So the organic dyes, if we look at the chemical fluorophores, so organic dyes, you certainly recognize some of these names. So these are used to label, for example, an antibody, but you can also have specialized markers for certain compartments or certain molecules, for example, DNA, like Hoechst and DAPI. But we also have special organic dyes for membranes, cytoplasm, mitochondria, and they can be used in fixed or live cells. Some of them are specialized for live cells, some cannot be used in live cells. So we have a wide variety of organic dyes that can be used that are chemicals. And one of them, as an example, we have ratiometric dyes as well that change excitation or emission spectrum depending on the pH, for example, the calcium concentration. So this is an example of, oh, okay, it's, I don't know if you can see it, but anyway. Now. Right, so this is an example of a flip calcium 5 in neurospheres where we can see, so where you have a high concentration of calcium, this is when the fluorophore is going to fluoresce. Where you have a low concentration, there's a lower fluorescence. So you can measure the calcium concentration with this dye. This is very useful. Another example of chemical fluorophores are those that blink. And you can see in here, this is used in storm super resolution and palm as well. And these fluorophores are simply different because they're prone to going into the dark states. And now you know what it means. It means that they are going to be excited and then they become dark. And then later they come back and they start fluorescing. And this is why they're blinking. And then they make very many cycles of this. And so alexafluorose 647, for example, is very good at that, attofluorose 655. So this is used in super resolution. Now, if we look at the protein fluorophores, so they are produced by the cells. So we make a plasmid and we fuse our protein of interest with the cDNA of a fluorophore. And then we transfect the cells, and then the cell is going to produce this modified molecule. It is very good to follow specific molecules in live samples. For example, the problem is that it requires overexpression of a highly modified protein. So that is not a natural state of affair for the cell. And so artifacts are very common, estimated around 30% of all furan protein. And this is the paper, if you want to read. But they are often overlooked. So we can have problems with the maturation of the protein, for example, how it folds. We can have problems with localization. We have problems with interaction. It is very important when you make a fusion of protein to be sure that you are you're testing different fusions and you compare with what normally the way the protein behaves and where it's located to detect these artifacts. Fluorescent proteins come in many colors, and this is evolving constantly. So the original fluorescent proteins were extracted from jellyfish and a bit later from corals. And then they're having derived and derived and derived. And now we have all sorts of colors. And every year there are many new fluorophores that are created, that are brighter, more stable, etc. And it's important to check the latest. And the most development is into the red, far red, near infrared. So there's more and more because it's less damaging. So more and more fluorophores in there, and that means we have a wide variety in these ranges also of spectra. So it's important to find the correct things that work for you. We will talk about this later as well. And then we have, some examples of specialized fluorescent proteins. For example, the photoswitchable, which go from one color to another color, and photoactivatable, which goes from dark to color, so on and off. You can turn it on and off. For example, Drompa and MKAD. So this is an example of MKAD in the zebrafish, and it's transgenic, so the whole fish expresses MKAD and it's green. But if we shine some light on the nose in here, we're going to see that these molecules will become red, and then we can follow the cells that have been exposed to light throughout the development of this fish. So here we go, we turn on MKAD to red, and now the cells are moving around in the fish, and we can follow the cells that were exposed to light. So very useful tricks with fluorescent proteins. Another trick is bimolecular fluorescence complementation, where we're going to basically split into a fluorescent protein and attach each half of the fluorescent protein to a molecule. When these two molecules are interacting, then the two halves of the fluorophore come close together, and then we have basically fluorescence. So this is a way to show protein interaction, or at least that the proteins are very close to each other. Another thing that we can use with fluorescent protein, which is very useful, is Thirster resonance energy transfer, or FRET, which is sometimes wrongly called fluorescence resonance energy transfer. This is wrong because this is not fluorescence. So what happens here is that we have it's not a fluorescence transfer, it's an energy transfer. So what we have here is we have a fluorophore and a second fluorophore, and each of them is attached to a protein. When these two proteins come close to each other, the two fluorophores come together. And what happens is that this guy, we're going to excite this guy with its own excitation light. And so it starts vibrating, it's in the excitation signal state. And what happens is, because the other one is so close to it, the energy from the CFP in this case is going to be transferred to the YFP, and the YFP is then going to be in the cingulate state, excited, and it will fluoresce. So we're shining light to excite CFP, but it's YFP that will fluoresce. So it is not the case that CFP is fluorescing and that light is exciting YFP. That's not the way it works. It's an energy transfer. CFP will not fluoresce, and YFP will not be excited by light. It will be excited by the energy that is transformed from CFP. So this is FRET, and it's also very useful. And here we have this little movie where we have FRET biosensors for histone methylation, so we can detect which histone is methylated with the colors that we get with FRET. So we can have, for example, a high level of methylation is going to be in red, the fluorophore is going to fluoresce in red, a low level of methylation is going to be blue, and then the intermediate is green. And when we look at it like this, we can see that in these cells, when they divide, the nucleus is going to become red because of the high methylation level. And then as the cell continue into the cell cycle, we go back into green, blue and green. And so with this, we can follow in live cells the methylation of histones. Sorry. So this is another example of these tension sensor on focal adhesions, where you can see that we have one molecule with two fluorophores, and when the molecule changes conformation, then the two fluorophores come together, close together, and then there is fret also happening. And thanks to this, we can follow the focal adhesions, and you can see how they change colors as so the tension of the actin pulling on the focal adhesion is actually encoded in the color here. Something that we spoke about also tiny bit yesterday is FLIM, because we have one student who is this year who is using FLIM on the Leica system. So as we said, as I said yesterday, fluorophores have an excitation spectrum. They also have an emission spectrum, and everybody's aware of this, but they also have a lifetime. So the lifetime of the fluorophore is a specification of the fluorophore, and each fluorophore has a lifetime in the same way that they have spectra. This lifetime is in nanosecond, and the way it works is that we're going to have a special laser which sends a very, very short pulse of light. So we send this very tiny pulse of light, and then the fluorophores get excited and they start fluorescing, but now there's no more light, so they will slowly stop fluorescing. And so we can use special detectors to measure very precisely how fast the fluorophores stop fluorescing after the pulse of light, and the shape of the curve is the lifetime of the curve decay is the lifetime of the fluorophore. So you can calculate with this in nanoseconds the lifetime of the fluorophore with this calculation. And so this is very useful because the lifetime of the fluorophore is going to change with the environment. So when we have a fluorophore, some of them change with the environment, but most of them will keep the same absorption spectrum and emission spectrum, even if the pH changes, for example. Or if they have an interaction with other molecules, but the lifetime will change. And so then in this case, you can measure the pH or you can measure the interaction with other molecules. And so this is also a little example where you can see all these, I think that was a calcium pulse or something like this. So you will see that it comes, then suddenly the lifetime changes. So what we're imaging here is not the fluorescence color. What we're imaging here is the lifetime, and it's just color-coded to show different lifetimes. So this is FLIM. And a combination of the chemical fluorophore and the protein fluorophores is the hybrid techniques. So we have lots of tagging proteins that are very, very convenient, because the problem is that many protein fluorophores are very big. If you want to label a very small protein, the size of the fluorophore is going to be significant, and it may interfere very strongly with the function, the normal function of the protein you want to tag. And therefore, it's much better to have a tiny modification of the protein. So in click chemistry, SnapTag, flash rehash, or genetic code expansion, for example, are techniques that use this hybrid system where we have both a protein that is overexpressed and a chemical. So we overexpress the protein with just a tiny modification. In general, it's just one amino acid. And then we add to the medium a cell-permeable reagent, so this is a chemical, that binds to this modification. So it's highly specific, and there is a low disruption of the protein conformation due to the fluorophore. It's very low distortion. But anyway, we require overexpression. So we can have the same type of artifact that we get due to overexpression of a protein. And I think this is my last slide. We want you to pick the best fluorescent protein. There is a fantastic tool for this. It's called fpbase.org. And if you're going to have a little exercise later on where you're going to go and explore, and you will be guided to explore and find the best protein for you with this website. When you need to think that when you change the fluorophore that everyone has always used in your lab for the latest fluorophore, this may dramatically improve your results. Fluorescent proteins and dyes evolve all the time, so do not use just the stuff that you have in the fridge just because it's in the fridge. Do your job in finding the latest, And then you can really see dramatic improvements because fluorescent proteins have improved a lot. So this is what FP-base looks like. And again, we will guide you through it, but you will see each of these little dots is actually a fluorescent protein. You can get the excitation emission spectra. You can pick a new protein that matches better your fluorophore, your microscope, for example. You can get to see the lifetime of the fluorophore in this website. You can see the maturation time, which is how long does it take for the fluorescent protein to actually fold and be ready to get out of the ER. You can see the photostability, for example, how fast it bleaches. And the extinction coefficient is the capacity to absorb light. So if I send 20 photons to this molecule, how many does it absorb that can excite it? And the quantum yield is the opposite, is that what is the proportion of these photons that are then going to be spitted out as emission? So quantum yield. And the brightness is, these two are multiplied together, and that gives you the brightness. So you can see all this in this, and you can pick the brightest fluorescence protein for yourself. The oligomerization is also very important. So we have under this table in the website, we have something where you can select to only use monomers. And that is very important because even weak monomers are likely to disrupt the way the protein works. So you're binding a protein that normally does not dimerize to fluorophores that do dimerize. Clearly, the fluorophores will bind to each other, and then the two proteins will be close to each other. That will affect the protein function. So you want to use only monomers. So you should go and check if your fluorophores are monomers. And this is especially true if the protein of interest is concentrated in vesicles and membranes, that you have a higher risk that the fluorophores come together. You also want to pick the best chemical dye, and that can also change your result dramatically. This is exactly the same, except that we don't have oligomerization and maturation time. These are only for proteins, but this is exactly the same for the chemical dyes. Unfortunately, there is no nice database like FP-base, but this book called the Molecular Probe Handbook, you can still find it in the Thermo Fisher website, and it has really a lot of information about the different dyes. So you can find here the latest dyes. One little word about autofluorescence. It is also part of fluorescence. So autofluorescence is real fluorescence, just is the one that you maybe do not want. This can be dealt with spectral unmixing, which we will talk about later, with chemical treatment and bleaching, and this will be also in the sample preparation lecture. And in some ways, sometimes it can be an advantage because you can see your fluorophore without labeling it. So you can get label-free imaging with autofluorescence, and you should think about it in that way that maybe you can find an advantage to it. So all this we will deal with in the sample preparation module. And here you can see an example of heart tissue treated or not treated with Sudan black to remove the autofluorescence of parfumine. Okay, so in conclusion for this little lecture, please choose bright and photostable and monomeric fluorescent proteins. You can look at them in FP-base. Check that you have efficient excitation and emission detection on your microscope so that things work. This is what we're going to do this afternoon, so that you're imaging efficiently. And pick fluorophores that have the lowest risk for bleed-through on your microscope. That's also what we're going to do this afternoon. And ask the company for the best chemical fluorophore for your application. Do not hesitate to write to them and say, I would like to do this and that. What is your best fluorophore for this? Because things evolve all the time. Do not pick a fluorophore from the fridge just because it's there. Okay? Try to improve things. So I am now finished with this part of the lecture, and I think the